Welcome. Today we are focusing on telomeres and all their various glories, ranging from substantive effects to sort of validity and reliability information that probably we all should have collected years ago, but better late than never. So without too much further ado, I'm going to present the first set of results from the Telomere Research Network presented to us by Stacy Drury for the next 15 minutes or so.
All right, and since this is a group that stays on task, I'm gonna try to be super quick. I'm assuming everybody can see my screen. I'm just gonna go with that assumption.
Looks good.
All right. So first of all, just an introduction to the Telomere Research Network. This was actually an RFA that grew out of a telomere meeting that was organized by Liz Nielsen and several others to really talk about some of the methodological concerns for telomere studies. And I'm going to be very clear that these are telomere studies that are specific to population-based human studies. So, and during that sort of expert panel meeting, there were several identified critical areas relative to measurement that I expect that this biomarker group has spent a lot of time talking about. Sort of how do we really think about establishing protocols and parameters for the most scientifically rigorous measurement of all biomarkers, right? So this is not an unusual occurrence or challenge. And I think one of the things that has happened is that as we develop new technologies, particularly ones that are translatable to multiple different types of laboratory setups, the speed with which there is uptake of the methodologies often is far more rapid than the sort of really thinking about the rigor regarding the procedures and methodologies and quality control over those measurements. So people uptake different techniques. We get a lot of data really quickly. It's an exciting field. And then there's this sort of recognition that we need to pause and really go backwards and think about the precision of our instrumentation. And so that is sort of what created this collection of different grant opportunities. So the structure of the TRN is that it is a cooperative agreement. So we have wonderful intramural, or we have wonderful NIH partners with NIEHS and NIA. There is a U24, which is sort of the organizing structure, which I think very much overlaps with what is happening with the Biomarker Network. And then there are 4 U01s that are independently funded to both focus on the sort of methodological components and cross-lab studies, but also have their own specific aims. And so I'm going to talk more about the methods comparison study and what we're talking about doing there, but just sort of a general overview of the 4 U01s, and these are very general descriptions of their overall aims. Is one is looking very much at pre-analytic factors, particularly focused on how pre-analytic factors from the very beginning of collection all the way through to the right when you put it on the plate influence PCR-based TS ratios and telomere length measurements. A different U01 is looking at the cross-tissue correlation. So as with all sort of epigenetic studies, we often know very little about the cross-tissue correlation of measures. So this process is something that still is ongoing for methylation, and certainly about developmental differences, right? So we don't think any of these markers, particularly telomere length, is static over the course of development, but we also don't think that the change in telomere length is static over the course of development. And to date, there's too little information about that, particularly on the younger end of the life course. There is also a U01 that's, in addition to sort of helping with the method comparison studies, is looking at the development of a new rapid throughput assay based on QFISH. And then one that is looking at sort of telomere length as a predictor of outcomes in cancer treatment and transplant. So really trying to establish the utility in a clinical realm. So the U24, the goal of it is to really coordinate the U01 efforts and incorporate into the sort of direction of future research the results from the method comparison studies. And I underlined this, experts from the telomere field. Oops, sorry. Whoa, that was interesting. To really expand beyond the original people that were involved in this and make sure that we are integrating input from not just people focused specifically on telomere research, but people outside of our field as well. For the methods comparison, we really want to establish the importance of biologic sample collection, storage, and processing. And I imagine for those of you who have ever worked on something like cortisol, we've had these same conversations in terms of the challenges of really establishing what are best practices. And then laboratory methods and data analysis and reporting requirements. And the sort of first product of the TRN, I'm hoping will be submitted as a peer-reviewed paper, but certainly posted on the website in the next month, is really reporting requirements for PCR-based telomere studies. So another reporting recommendation that's out there. And the last part is the sort of future directions, which is develop and foster a network that really is focused on understanding telomere length as a marker of psychosocial and environmental exposures and a predictor of disease. So once we are confident with our methodology, moving forward to really say, what are the implications of telomere length? And how do we best utilize this biomarker or this measurement of cellular aging or stress to be useful for both measuring cumulative exposures, but also predicting disease outcomes. These are the people involved, and I know that several of them are on the call now. The U24 is myself and Alyssa Eppel, John McLaughlin, he used to be one of the scientific directors at NEHS, and Simon Verhulst, who puts up with me a lot because he is the analytic genius behind everything. Our NIH partners, so Liz Nielsen, Max Gao, and Michelle Heacock, and then the PIs for the 4 UO1, so Abram Aviv, Julin, Idan Shalev, and Ling Zhang. We do have an external advisory committee, so some of you will see a familiar face on here and hopefully recognize our amazing external advisory committee that really helps make sure that we are moving in the right direction and helps us also identify other people, some of whom I have seen on this call, to tap to be involved in the network as we grow. Really important for me in taking on this responsibility was that there are 2 really critical guiding principles for what the network will be doing. And the first is transparency. So we will be putting protocols up and really trying to cultivate and curate data so that people can replicate and really make sure that we are improving the rigor of the field, and then really providing guidelines about reproducibility and replicability for any measurement of telomere length. And I think those are critical to advancing the science. Goals of the network really involve 3 components. So research, so really making sure we have the information needed to establish best practices. We will talk a little bit about pilot studies and what the network is able to do. Outreach and dissemination, so this is part of the plan. It's a little bit different than we anticipated. So trying to have conversations across different scientific fields at scientific meetings, highlighting seminal papers, which you'll find on the webpage. And then the short and long-term outputs are really technical guidelines, best practices. And then towards the end of the first 5 years, hope we'll be able to have in-person methods workshops. So really helping people do hands-on learning for different methodologies. So in terms of the method comparison studies, this is actually somewhat ongoing. We have to get most all of the labs reopened before it will be officially ongoing. But really understanding what drives differences in telomere length assays performed in different laboratories. Development of best practices recommendations for assays. And this is not just going to be for PCR-based assays, but really the hope is to devise best practices recommendations and reporting guidelines for all of the different ways that people are measuring it that can serve as a template for future methods as they are developed. And the last part is to enhance existing methods or support the development of new methods. And I put this part at the bottom, which is that we really are charged with understanding telomere length measurement for population-based health research. So this means that we have to take into account the caveats of the collection of biologic samples at that level of population-based studies, as well as the challenges with creating throughput that makes sense. And again, we're not reinventing the wheel. I think this is something that has happened repeatedly. So for things like sequencing, for expression studies, for DNA methylation, all of them kind of go through this process. We're just working to catch up with telomeres. Okay, sorry, it's really, so why join forces with the biomarker network? First, I think much of these conversations you guys have had for a long time, so I hate reinventing the wheel. So hoping to learn from the things that worked and the things that didn't work, as the biomarker network sought to advance some of these same goals for other biomarkers. Really understanding the relationship between telomere length and other biomarkers. So there is some data out there, particularly I would say with cortisol. But again, with a lot of the early data, it's not always super consistent. My dream is to develop integrated methods of the collections of biological samples that allow us to truly measure biomarkers, multiple biomarkers within the same sample, because I feel that strengthens the validity. Explore the opportunities in existing studies where we can add in telomere length. So one of the things about the network is that there are funds to do additional studies that can be done at other locations. So really thinking about what types of cohorts or studies that exist within the biomarker network where there is the feasibility of adding in the measurement of telomere length to provide robust information about how telomeres relate to other biomarkers, or how both the sort of separate predictability of biomarkers or the integrated predictability of biomarkers may help us better understand how different exposures predict disease, or how we can predict disease outcomes as well. So how can you get involved? So first, we are forming subcommittees to really target and identify the important questions that face the telomere research field. So some of these involve sort of looking at the relationship between telomeres and biomarkers. So we have a biomarker committee, which I think is most relevant here. Another committee is going to be focusing on early life measurements. So what we know about the change in telomere length that is expected over time in kids. So that form is a Google form that you can self-nominate, or you can nominate somebody else. The sort of thought process with the subcommittees is these are great opportunities to really expand your own knowledge. The expectation is that these committees will present recommendations for white papers and published papers, but also can present important scientific questions to the network and potential pilot projects that the network would be able to support. So really taking a deep dive into one aspect and saying, hey, this is a big important scientific gap that is critical for advancing the field, and presenting that to the U24 as we have funds to fund pilot studies. We do have a pilot award. This is more designed for early investigators, and it includes a $4,000 honorarium that also has mentoring analytic guidance as needed. We've extended the deadline to July 15th. And so if you email the telomerenetwork@gmail.com, I will send you the RFA. These are really designed to do secondary analysis of existing data to answer important questions about how telomeres are related to different environmental exposures. But the goal of these is to really bring in and support early career investigators, and to have them develop projects that can lead to subsequent either Ks or R01 applications. Other ways to get involved. So I am going to schedule, and I just picked the time, July 23rd at noon, a Telomere TRN public webinar where we're really going to talk about what the network is doing currently, sort of highlight the webpage and all of the different components to it. Again, if you want to join this listserv, please do. And then the last part is to become a partner laboratory to participate in the method comparisons and pilot studies. So while we have the U01 labs, we are hoping to really expand this to get other participants. Oops, sorry, I don't know what happened. To get other labs to participate. So we want to build the repeatability and reproducibility of all the different assays by really demonstrating that these things can happen at different levels. And so with that, I will take questions if we have time.
We do indeed have time. So folks, you can either raise your hand and ask your question in person, or you can submit them using the Q&A feature at the bottom of your screen.
So Steve, there's a note, someone asking, could you please place the subcommittee link on the chat?
I am happy to do that as soon as I get out of the screen share. All right.
And Eileen, I'm not a co-host, so I don't think I can see people raising their hands. So if somebody else
I don't see any hands raised right now.
Okay.
Does anyone have a question?
I don't know how to raise my hand, but I can talk.
Okay. Okay, Dan Notterman.
Yeah,
You
hi.
can talk? No way. Let's see.
Stacey, thanks very much. I want to compliment you and your group for first of all, successfully applying for this grant, and second, for the work you're doing, and I hope organizing all of us. It's really, I agree with you. It's essential work, and as you continue over the next few years, the sun will rise and the fog will dissipate, and we'll all have a better understanding of telomere biology. I was wondering if you have a chance, could you mention some of the methods, comparisons you've been working on? I realize COVID stymied a lot of that, but where are you headed with that, and what have you been working on?
So the first two are really focused on pre-analytic concerns. So the sort of steering committee, which is the UO1s, the U24, and our NIH partners, recognize that that has been, we think, the biggest challenge. So the two first studies that we are hoping to implement as soon as everyone's labs are open, one is a relatively large, completely blinded cross-laboratory comparison of the impact of DNA extractions from whole blood samples. So this is really going to be taking the most common utilized DNA extraction methods and integrate multiple different PCR-based and TRF-based assays to look at the inter and intra assay reliability for these methodologies, because we really believe that this is a critical and often missed opportunity to strengthen the rigor of, and I would say PCR-based studies have taken the biggest hit, but I firmly believe it has implications for other methodologies that extend beyond this, and so we are going to be really focusing on that. The second one that I think is applicable when we think about the utilization of storage specimens is that we will be working on one focused on understanding and again, in a very blinded and rigorous fashion, the impact of DNA degradation. So there are small studies out there, and we all have our own thought processes. What I have learned over the last year with really delving into the literature is that to really answer these questions, we need blinded, adequately powered, very controlled and very focused studies. And we need to know what the implications are of DNA degradation because there are thousands and tens of thousands of samples that are stored. And I know the Biomarker network has lots of those cohorts, and we want to understand if it's feasible to use those. The most sort of on the front coming out part is actually a critical systematic review of all of the cross-method comparison studies that involve a PCR-based method and any other method. So I think there's a lot of lore out there about what we know and what we don't know about both the sort of repeatability of the different assays, but also the comparability across assays. And so what this has shown is that we really don't know that much, so that there's a lot of insufficient reporting of what people are doing in their assays, and certainly insufficient reporting of what we believe are critical parameters for repeatability and reproducibility. So those are our three sort of initial targets. And I think over the next 6 months, we are going to be really also looking at trying to do some comparison studies for clinical implications. And I have a feeling that the COVID pandemic may provide us a very unique opportunity for that in terms of saying how can we understand sort of the relationship between an immunologic disease that seems to be implicating or very correlated with many of the conditions that we know are meta-analytically related to telomere length. So obesity and diabetes being sort of ones that seem to have a greater potential for higher morbidity and mortality with COVID. So that was a long-winded answer, sorry.
That's great. That's very exciting. And I think that we will have a lot of things to talk about together, our groups. Great.
Sounds good. We should move on to the next talk now, you guys.
Okay.
We are going to have a mashup, actually, of two different groups that have been working together to answer some of the same kinds of questions in the context of the, sort of biodemography centers and their interests in, sort of accurate metrics on telomeres. This is going to come from the team of Jude Carroll, Dan Noderman, Coulter Mitchell, and Jessica Fall, who talk about telomere length estimates, lab-to-lab comparisons, and sample handling effects.
All right.
Who's putting the slides up?
Jude is sharing.
Right. So can you put that on? There you go.
I think she's trying.
Thanks. So hi, I'm Dan Noderman from Princeton, and my colleagues on this talk, nicely introduced by Steve, are Jude, Jess Fall, and Coulter Mitchell from Ann Arbor, and Lisa Stepper from Penn State and Princeton. And we're going to give a summary of where we've been and where we are, looking always for collaboration and learning from each other. Like many groups, we've been stymied since March, so this is definitely a report in progress. And we call ourselves the LA2P Telomere Consortium. That is a great moniker supplied by Jessica, and if anyone has a suggestion of how we should rename ourselves, that's great also. Next slide, please. So I'm going to begin the talk, and then I'll be turning it over to Jude and the others. So who is advancing this for me? Steve?
Jude.
Jude. Okay, Jude,
There
you're
it
up.
is.
Okay, so we can skip through this. It's clear that interest is strong in telomeres, and we completely agree with the point that Stacy enunciated, that measuring telomere length accurately and reproducibly is foremost in everybody's interest, and maybe it hasn't been done in the recent past. Next slide, please. Okay, so I just want to take one slide. I actually pulled this slide out of a course I teach at Princeton, the Introduction to Cellular and Molecular Biology, just to remind people that we're going to be talking, at least in our conversation today, about qPCR as an analytic measure for telomere length assessment. As Stacy mentioned, there are many others. There are several experts on this call in other approaches. And of course, what we're measuring with qPCR is not the length of anything, but the mass of DNA that is reacting to specific probes, either to probes for telomeric DNA or to probes for some marker or representative gene. So obviously in qPCR, the basic principle is that PCR is used to amplify a specific region of a gene, and it amplifies it exponentially, doubling with each cycle. qPCR is an adaptation of Kerry Mullen's strategy of PCR, in which the reaction takes place with fluorescent reagents, a variety of different fluorescent reagents, and some kind of detection device that enables us to watch as the application proceeds in real time. That's why one of the other names of this technique is real-time PCR. Can you see my marker, or does Jude have control of the marker? Can anybody? Okay, so just looking at the right panel, the top panel on the right, you see a single analysis that's being measured by RT-PCR. And you'll see that at the very beginning, where the cycles are 0, you see virtually no fluorescence. And actually, for about the first 18 or 19 cycles, now remember, we're watching this through some kind of optical scanner. There's no accumulation of fluorescence. There's no incorporation of the dye. And then gradually, and then more rapidly, you see the fluorescence line increase until it begins to increase in an exponential way. At that point, you see it crosses a threshold, which is an arbitrary threshold, which is assigned CT, or cycle threshold value. As the PCR reaction continues, it enters a non-exponential phase. That exact point is often above 40. It depends on the supply of reagents, basically, and other factors. But after a while, the reaction is no longer linear and can't be relied on. So the most important point to remember with qPCR is that the number of cycles in which the PCR product exceeds a threshold value, here CT, is inversely proportional to the starting mass of DNA. So in general, the less mass of DNA that is the target of your primers, the more cycles it takes to visualize it or for it to transit past CT. And so that's shown on the bottom graph, where there's a standard curve put in. And the leftmost curve denotes DNA which was added at a relatively high concentration or high mass relative to the others. And then the second at a bit less, and the third at a bit less, and then the fourth well had the least DNA to start. And if you graph that, that's a linear relationship between the input mass of DNA and CT. And you can see where these curves cross the CT threshold at successively different numbers of samples. So we're going to be basically looking at 1 over CT. And as the CT gets higher, the input mass is thought to be lower. Now, the number of different DNAs that can be measured by this technique at one time is limited only by the number of different primer pairs you can figure out, and also the instrument that you're using, how many different lasers or filters or whatnot that you have. Okay, next slide. So this is actually a screenshot from an ongoing measurement. Again, I'm sure many of you are familiar, but just to make sure. In telomere length qPCR, we set the primer pairs to hybridize to both telomeric DNA and to a reference gene. 36B4 is generally used as a reference gene in these operations, although there is a recent literature suggesting that the underlying assumption that this is present in one copy in human genomes is not always correct. We continue to use 36B4 as a reference gene. Other laboratories do use other genes, and that's something to think about. We detect the fluorescence signal that's related to the y-axis here. We use cybergreen in this. It's an intercalating dye, which as DNA forms duplexes and the mass increases, accumulates more of the cybergreen. And so there's a decent relationship between the mass of the target DNA that's being created and the overall fluorescence. As Stacy hinted, we can report results either as a ratio of intensity or as a ratio of CTs, as the TS ratio, which is the original way of doing this. And then it's also possible to report results as an absolute PCR, where you include a standard curve and can interpolate the mass of DNA and hence the number of kilobases or the number of base pairs of DNA that you assign to the telomere probe, so that we can get a readout in kilobases. It may be that the TS ratio provides less variance. I'd be interested in seeing what people think and what their experience is. And in my lab, in general, we report kilobases. And this is just an experiment where we've obviously had a number of DNA samples. Some of these are unknown samples that we're measuring in one of our studies, and others of them are a standard curve that we use so we can interpret the TS ratio in terms of kilobases. And you see that all the way on the left is a sample which had probably lots of telomeric DNA, and on the right are ones that have less. Okay, next slide. So I'm going to recapitulate a meeting, some results that I think Jessica talked about at a previous biomarker meeting, just to show you what our thinking has been and how we evolved to this interest. This was an experiment in which we collected saliva in Genetech. I think there were 505 kits from 30 volunteers. And we collected the saliva by having them spit into a 50 ml conical tube and immediately dividing them into origin saliva kits and mixing with the solution. And we put those basically in storage. The DNA from the first kit times 0 was isolated and stored in a RefCo at -80. And then the remaining aliquots stood at room temperature for 1, 2, 6, or 12 months, after which time the DNA was isolated and frozen. So now we have 6 samples over a year's period. They've stayed in the origin solution for the times that we let them, and then they were stored presumably stably at -80 until they were all analyzed together. Next slide, Jude. So one result that puzzled us and made us unhappy is that we saw a sharp decrease in double-stranded DNA yield between 6 months and 12 months of storage. And unfortunately, I wasn't prescient enough to put in an 8-month sample. So we just saw it be stable for 6 months and then disappear by 12 months. So the DNA yield decreased from about 39 micrograms per sample down to about 2 over that interval. Next slide. As you might expect, we also saw a decrease in the measured telomere length between 6 and 12 months of storage. Again, we don't have an 8-month sample, but it was a significant decrease and probably a meaningful decrease. Next slide. And when we looked around to explain this, we performed gel electrophoresis upon thawing these samples. And you can see that there's a large size DNA, as you would expect. In the first sample, there's some degradation, but there's large size DNA. But pretty quickly, in this one, this is one person who we call S16. It's not a ribosome. And you can see pretty clearly that the DNA is degrading over the period of time until it's hard to find in the last sample, in the last example of this sample. And of course, I'm selecting one that makes the point I wanted to make. All of the samples didn't degrade like this, but enough did to pull down our results. Next slide. So once this happened to us, I showed this to you because this was sort of the incitement for Jude and Jessica and Coulter and Lisa and others that we're working with to do also what Stacy's been talking about, to begin to take a step back and think about what we're really doing. And one of the first things we did was to create a scratch pad of all the things that can go wrong. And I'm sure that the 74 colleagues of us that are on this call right now have thought of many other problems. And I'm really excited to have this conversation, to hear what you're working on and what you've thought of also. Clearly, as Stacy mentioned, the source of DNA has to be considered. I showed you a saliva result. I'll show you some preliminary data from a blood result in a minute. How the blood is collected. Are we using PAX genes? Are we using EDTA? Some people still collect the heparin, that sort of thing. What are the storage time and temperature considerations? And another very important variable that I'll get to in a minute is the cell distribution, and how the cell distribution is affected not only at the moment of phlebotomy and by concurrent conditions or diseases the person might have, but also by the process of storage. As Stacy mentioned, the extraction method is key. The method of suspending the cells or resuspending the cells is key. That's part of extraction method. Storage time and temperature. Jude will show you data about freeze-thaw cycles in a moment. And even how we're quantifying the DNA. Are we using something that quantifies double-stranded DNA only, something that incorporates fragmented or single-stranded DNA? There are lots of problems with telomere measurement, and no method is perfect, as I think everyone agrees. We tend to measure or report absolute rather than relative qPCR results. And it may be that that's a mistake. It may be that that increases our variance. The reagents that are used, the tack that's used. Jude also has some data on the specific instrument that's used. And of course, the analytic protocol, the reference gene, and even the operator. I've noticed in my lab that some operators get consistent results and others maybe don't. Go on, please. Okay, so after that study that we showed you, we recommended that saliva sample storage duration at room temperature should probably not exceed 2 weeks to a month, even when it's in a so-called stabilizing kit, pending further data, and regardless of what anybody's kit manufacturer tells you. And as an interim measure until people in our group and people in your group and others are able to give us perhaps more robust recommendations, it's our practice to try to measure DNA yield when we get the sample, and then just before we do the analysis, and if we see a substantial decrease in DNA yield, we tend to put that sample aside. And my rule of thumb is 50% of the original yield, but I acknowledge that that's not really a well substantiated criterion. Next slide. Oh, so the projects that our little group is working on now include repeating the stability test with extension to blood. I'll show you a little data. We don't have much. We have also starting to work on testing the collection tube at the same time. You'll hear some data on freeze-thaw stability and perhaps some data on lab direct comparisons between 2 different labs. Other projects that have been funded and that we've held in abeyance include a dried blood spot stability test over about 25 years of storage that we're doing in conjunction with the Department of Health. We have IRB approval and department approval to do this. We just need to get the Department of Health to turn their attention from COVID, so that might take a while. We're doing saliva blood comparisons under conditions of social adversity in conjunction with the Geronimus group at Ann Arbor, and this paper is under review now, so I won't talk about the data. And I believe that I've just been funded or will be funded for a large study, about 2,200 people, to look at DNA methylation and telomere length in age 22 to 24. It's a national sample that will also include cardiovascular health measurements. And perhaps we'll have some preliminary data to talk about next year or the year after. Next slide. So the first project we started to do before we were shut down was to repeat the stability test that I just showed you, because it was a funny result to us, and also extend it to blood. So we tested various methods of blood collection, you'll see in a minute, and storage conditions to inform best practices for these measurements. And this is oriented towards population-based type of research. And we replicated the saliva pilot. So next slide, please. So we collected whole blood in 28 volunteers using 3 different tubes each, EDTA, heparin, and PAXgene DNA collection tubes. The blood was stored at 4 degrees for a variety of intervals up to 28 days before DNA extraction and freezing. DNA extraction and freezing followed standard protocols we can talk about later. We also did about the same thing. We had parallel tubes in EDTA so that we could monitor CBCs because it's ultimately our intention to submit these specimens for telomere length measurement in a couple of laboratories and also for DNA methylation measurement, and to compare how cell distribution with storage affects these indices. Next slide, please. Okay, so as you might expect, by keeping these samples before submitting them for CBC analysis, we see changes in the relative proportion or the average cell counts in some of the constituents that I selected. So this was a clinical CBC. These were done by LabCorp, but I've limited it to the dominant nucleated species of lymphocytes, monocytes, and neutrophils and not including other forms. And you see that while the red cell count, the total white cell count, were pretty much stable while stored for 8 days, we begin to see changes in the lymphocyte count after about 4 days, 3.5, 4 days, the same for monocytes. And for neutrophils, there's a very wide variance. I'll show you the individual data in a minute. And just because of the wide variance in starting total white count, we've just calculated a ratio. So all of these measures are relative to the original count at 1 at 0 time. Next slide, please. These are the individual patients or subjects. Again, total white count, red cells, polys, monocytes, and lymphocytes. And you can see the counts are generally stable, but there are some in which there seems to be, this is all from EDTA, by the way, there seems to be a fairly pronounced decrease after 2 weeks. Others in which after 4 days, this is the sample number. Others in which in 4 days, the cells seem to disappear and reappear. My guess is that has to do with the cell conformation and the interaction with the cell and the laser leading the instrument to zero it out then, and then the cells change their morphology again, and then they're counted again. It's hard to make sense of this data until we also have the DNA measurements, the DNA quantity measurements, and also have a chance to interpret this in the context of the telomere lengths. Okay, next slide, please. Okay, so I'm going to turn this over to Jude now. Jude, you here?
Great. Everybody hear me?
Yeah, go ahead.
Fantastic. So the next project was the freeze-thaw pilot study, where we wanted to test whether freezing and thawing the DNA after extraction would result in changes in our estimates of telomere length. So we designed this experiment where we collected fresh samples from 12 participants, extracted the DNA at that same day, and then aliquoted the DNA into 3 different tubes. All the tubes were then placed in a negative 80-degree freezer. 2 of the tubes were then removed and thawed and then frozen again. And then on the 3rd day, the F3 sample was removed, thawed, and frozen again. And then we had 3 freeze-thaw cycles for those samples. So one had 1 freeze-thaw, one had 2 freeze-thaw, and one had 3 freeze-thaw. We pulled all of those, thawed them all together, and ran them all on the same plate so that the results were comparable. What did we find? So our initial results, you can see here in the figure. Overall, there was no significant differences in the telomere length estimates between the freeze-thaw cycles, which are, you see 1, 2, and 3 freeze-thaw cycles. Note here, this is, there's some trends towards declining, although that our t-tests were not significant for the mean differences. So we had just finished this up really when COVID happened, and so we wanted to think about our next steps going forward. One of the things that we note is that the sample size is a little small, and we might be able to make more inferences about freeze-thaw cycle number if we increase the sample size, increase the diversity of telomere length. So you'll notice that a lot of them are in this below 1 range. These are from older adults primarily. So we think if we recruited some individuals that are younger that might have longer telomere length, we may have more vulnerability to the freeze-thaw cycles if in individuals with longer length. So we wanted to look at that next, and then possibly continue this out. So we're not really seeing significant differences in telomere length estimates with 3 freeze-thaw cycles. Might we see it at 4 or 5 freeze-thaw cycles? So those are sort of the preliminary findings for that. So next, the project 4, we wanted to see if we could have two independent labs run the same telomere length assay and get the same results. So we had Dan Notterman's lab at Princeton, and then my lab at UCLA called Aging Biology Behavior, or ABB. Used the same assay and the same samples. So we used Dan's assay. We got it set up in my lab, and we had samples from my lab shipped to Dan's, and then samples from Dan's lab shipped to us. And so we ran all of those on plates at the same time. We made sure we wanted to use the same ingredients in the assay, so we're trying to be as consistent as possible. We included the same primers, ordered from the same source, the same cybergreen, the same standards, the same lot of reagents, not just the same reagents, but the same lots. Each lab ran the assay using their machines. At my lab, we have a Biorad machine, and then in Dan's lab, they have the Stratagene. So the question was, do we get similar telomere length estimates? First, we looked intra-lab, so within Dan's lab on the left, we see a high correlation between their first and second run. Similarly, in my lab, the first and second run, the ABB lab, has high correlations, so intra-lab correlations were good, although you can note that the absolute telomere length estimates from this were quite different. So then we went to the direct comparison between labs. So we're running the same samples in two separate labs, and the correlation drops quite a bit. You'll notice that the absolute values, again, between the two labs do seem to vary, even though we were using exactly the same standards and estimates for the telomere length. So what we did then is we decided to decompose the two, looking at those CT or cycle thresholds for the telomere length and the cycle threshold values for the single copy gene, the 36p4. So the telomere repeat CT values, you can see for the standards, are off by about 3 cycles. So the ABB labs cycle threshold hits at 25 and the Princeton lab at 22. And you can see there's a consistently different CT values. But for the 36p4, they're almost identical. So really the point is that we think the 36p4 is running similarly, but we're getting very different values for telomere length estimate. And as a result, we get a different overall telomere length value. So then we ran an experiment just to check, if we had differences in our PCR machines and their ramp time. We noticed there was a slight difference in ramp time. The Biorad was quicker in ramping up to temperature, and so we adjusted the ramp time to try to match the Princeton labs machines. And so by doing that, we did not see a change in our CT values. You can see here from the old ramp time and the new ramp time within the ABB lab, the CT values are still the same. So that probably wasn't it. So the next thing that we wanted to investigate about the machine, thinking that that was our only variable really that was different between labs, was whether or not the thresholds were set the same. And it turns out that they are different. So there's differences in the way that the machines determine the threshold values. So the Biorad method is proprietary, whereas the Agilent chooses the fluorescence value associated with the lowest difference in CT and then sets the threshold there. And we're unable to convert between these two machines at this current time. We're still trying to work that out. Also, it's important to note that the Princeton lab did obtain a Biorad machine identical to the ABV lab, just to see if they ran the plates simultaneously on those two machines, on their machine and this Biorad machine in their lab, if they got the same results, and they did. So we are seeing clear differences in that cycle threshold on the Biorad machine compared to the Stratagene. So next steps include investigating this further, of course. One thing we need to better understand is the difference between the PCR platforms and see if we can make some recommendations that will standardize across labs when performing these assays. So there's a number of factors of the PCR machine that might be playing into this, including we have to look into the hardware, the software, the algorithms for the CT is one thing, but there might also be some key differences in hardware and how the fluorescence image is detected. We also need to consider, if we can optimize, can we make recommendations on best practices, maybe using a set of standard samples that everyone can use that we know works across different machines. Okay. So those are the two studies, sort of where we're at, and we're in progress on both. And then this is just a slide to kind of go forward, thinking about next steps across all of these different things. A number of things are in progress. The test collection tube Stan talked about, we can look at telomere length in the future on those samples, and then increasing sample size and diversity for the freeze-thaw study. The lab-to-lab comparisons, we're going to continue trying to identify really the main contributor to the difference in our estimates there, and then the projects that Dan had mentioned previously. Just want to make sure we acknowledge the support that we've received from NIA and ICHD, and a special thank you to Eileen and Teresa for encouraging us to pursue this work and helped build this collaboration. And then of course the Biomarker Network and the laboratory personnel in particular who worked diligently on these assays, including Lisa, who I think Lisa's on the call, Gabrielle, Lulia, and then Lillian Polsky, who's from my lab at the Cousins Center. I think we'll
Great,
open
thanks,
up to
you
questions.
guys.
Thank you. We have several questions in the chat. Michelle Haycock, can you ask your question?
Let's see, did that work? Am I off mute?
Yes, you are.
Yep.
Oh, okay, great. So I actually had two questions. So Eileen, I'm not sure which one. One was for the previous speaker, for Daniel, and the other one was for Judith that just spoke.
Go ahead and ask the questions, both then.
Okay, thank you. For Daniel, you were looking at DNA yield, and you said as a regular practice in your lab, what you're doing is you're disregarding the samples that give you less than 50% yield. And I wanted to ask whether or not you're including an assessment of DNA integrity. You showed what looked like an agarose gel, I was assuming. So I was also wondering if your lab has looked at alternative methods to assess things like yield and DNA integrity, so things that could be more high throughput.
Yeah, so thanks. So when we did the original fragile families measurements, we did not routinely assess DNA integrity, but we learned, and so in further large-scale analysis of telomeres, we are going to be using a bioanalyzer. That was actually a bioanalyzer, not a standalone gel. And so I think that's actually a good precaution. Rather than simply to measure DNA mass, but to look at signs of degradation and quality. And we're going to use a bioanalyzer for that purpose.
All right, do you want to ask your question for Judith now?
Okay, thank you. And thank you, Daniel. So Judith, I was wondering with thank you for, both of you for your great studies. For your, Judith, the research that you showed, you were comparing two different platforms to measure, to get the CT measurements. And what I was wondering is, have you thought about comparing another telomere length measurement, such as, the southern blot, the telomere restriction fragment, or even the flow cytometry method, to see whether or not, I understand you're not going to get absolute telomere length, but what I was wondering, would you be able to see whether or not the trend would track, worse or better with a different platform?
Yeah, I can answer that, Jude, if you want.
Yeah, sure.
Yeah, so yeah, that's a great question, and I know that there's There are several experts on the call who should also opine if they want. We do southern blotting in my lab. We did not do it in this case, mainly, frankly, because we were surprised by the result that we got. We don't expect that a southern measurement and a qPCR will be exactly the same, but it would be interesting to see if we can help use that to adjudicate it. And we have not done many experiments using human tissues with qFISH. We do use qFISH and are expanding its use in animal studies where we're looking at telomere length in tissues where we can get very good very good histology from the tissues. And I'd be very interested, I was actually very interested to note what Stacy's point about developing a new qFISH assay. That would be terrific. The trouble with the kinds of samples that we usually get in my lab, there's no cellular integrity. So cell sorting is not really a great option. But for these experiments going forward, we could certainly include those, and that's a good idea.
So Dan, if I might, just to add to what Stacy said earlier, that platform where she's working to see if she can get another kind of flow-based or it's a microscope technique, and it's not dependent on whether or not the cells you don't have to have cells that have integrity, because it's more of a hybridization-based technique. So stay tuned for that one.
Yeah, I'm looking forward to seeing that. That certainly would be, that would be great. We have thousands of samples to test it in if it's applicable. Thank you.
So there's another question in the chat from Waylon Hastings. Waylon, can you speak?
Yes, I can. Can everybody hear me?
Yes.
Yes.
Okay. My sample or my question was also for Judith. You were comparing the results across these two platforms. I was wondering if you tried running the plates through LinReg PCR to see if the CQ or baseline values generated in that program would be comparable across the platforms as a sort of added look, because it's sort of independent of threshold determination in that.
That's a great suggestion. No, we haven't.
Okay.
Yeah, thanks for that suggestion.
Yeah,
Cool.
that's exactly what we're looking for from everyone. If anybody else has thoughts.
That's what this group is for.
Jude, can you I had a question, sidebar from, if there are no other questions, just before we leave, I had a sidebar from Stacy. So if you can go to that slide I put in there, Jude.
That one?
Yeah, that one. I think, was this what you were asking, Stacy? You were asking about the ICCs. We don't have ICCs, but this was an experiment where in 132 or 99 people, we did repeated measures, three measures per subject in blood and saliva. And so you see the variances, and then we, over on the right, we calculated Cochrane's alpha.
Mm-hmm. Yeah, no, I was more trying to think about like where that was before you guys came up with the machine being the problem. But I think I was more trying to figure out if you were having issues because you were losing intra-assay reliability versus inter-assay reliability. And that was causing sort of greater variance in your TLS ratio, in your sort of absolute telomere length measurement. But it doesn't look like that's the case.
No, I think so. Our measurements are very consistent, whether or not they're accurate, but they're very consistent, and the coefficient of variation in the lab is, if you do a TS ratio, is under 1%.
Yeah, I think my concern was that the sort of DNA degradation issue may play a role in that. And I know this has been an issue with people trying to develop the droplet digital PCR-based assay in particular, which is going to be much more sensitive to sort of just variations at that level. But do we see greater intra-assay ICCs and inter-assay ICCs, even within the same lab, as we degrade the DNA. And my gut feeling is that we would.
Yeah, we have that data, and I could put it together, and we could talk about it in the future. That's a good point.
So Ben Williams, can you ask your question?
Hi, sorry, this is actually Karen Sugden. I'm Ben Williams logging. The question I had is that the groups headed by people like Steve Bustin have been using the Mike guidelines for qPCR for many years, and have had a lot of the same questions that are coming up now with telomeres. And I wondered whether you'd had a look at using that as a framework for trying to figure out where, you know, some of these differences between studies and samples and what have you are coming in. Things like, I'm thinking things like determining your PCR efficiency rather than just, you know, erasing everything to the power 2 and things like that.
Yes, we do that, and we do that routinely in all runs. And we don't always report it, and so I think we should probably report those parameters.
This is Stacy. I just want to say we actually love that suggestion, but I think there are some specifics to the different PCR-based studies. Say the Mikey guidelines are a great starting point for this, and to Dan's point, the sort of critical review of the literature, we as a field are not super great at reporting those guidelines, and so my hope is that this first white paper will come out. We actually have a checklist developed for this. You know, one of the things is to the Mikey guidelines standpoints, they sort of had empirical data that they sort of structured each of their recommendations would sort of give an indication of where the empirical data was. And I think one of our challenges for the field is that we really need to build the collection of that existing empirical data and fill in the holes so that we can say we're 99% sure these are important, but we don't really know what they do. And so my hope is that we can get that moving in the right direction. But yeah, I mean, I think the Mikey guidelines are a great place to start.
Thank you.
So there's
This
one
is
more.
Lisa. Sorry, I have one more comment regarding the PCR efficiency. We did compare the efficiencies between the Biorad and the Agilent, and we noticed that there was a difference. We generally keep our efficiency between 90 and 110%. And for the Biorad, we actually adjusted our primer concentration to improve the efficiency to get it within that range. And we noticed the same is true, that we had to change our primer concentration to get good efficiency on an applied biosystems platform as well.
Right, right.
Eileen, should we stay on time here?
Yeah, there's one more question. Shahinaz, can you ask your question?
Okay, yeah, I think I'm unmuted now, right?
Yes.
Yes.
Okay, so actually my question is related to the calibration. So everyone is excited when we can transfer something like T/S ratio into an absolute value. It's more meaningful and biologically easier to understand. However, it comes with its own challenges, you know, I mean, so I was interested to know if you have looked at how does this calibration function across telomere length distribution. So we have done a little bit of work here compared to flowfish in 200 samples where we have done, we have measured flowfish and qPCR, and we have done some calibrations. And what we've learned that it's not a linear relationship. You know, your agreement fall apart at the extremes, at the very short and the very long, and it was not really very, but the short and long where really the actions is. And perhaps this is what's causing what we see a lot of null associations using qPCR. If you calibrate or not calibrate, because it doesn't matter. I mean, calibration is only function of an equation.
Lisa, do you want to answer that? No? So we do not do a formal calibration, or at least we haven't. But I think that your question gets at a very important point, because as we've begun to talk to other investigators and work at other labs and look more carefully at our data, we're beginning to acknowledge that at the margins, especially since we're dealing with a ratio, we are seeing it may be that it's not smart to always look at the so-called absolute number, and maybe we should also be reporting the TS ratios at least as a backstop, because I suspect you're correct, and that the calibration is nonlinear, and it may also be not predictable at the margins. So I think that's a good point.
Good.
All right.
All right, thank you, everybody. Let's move on to the next talk now, which is more substantive results from the land of telomeres, demographic and developmental patterns in telomere length across adolescence.
Okay, great. Thanks to the organizers for the invitation, and thank you to the previous presenters. I think it's a really great sort of privilege to be able to go after everyone who goes over these very detailed measurement issues, and I appreciate that introduction as well. So keeping those measurement issues all in mind, what we want to talk about today is some descriptive findings around the demographic and developmental patterns in telomere length, and in a particular life course stage that is often understudied in this field. So I'm Lauren Gaydash, and I'm from Vanderbilt University, and I will acknowledge a lot of my collaborators who are also on this call a little bit later in the slides as well. Just a brief introduction, and by way of background, this audience really needs no motivation for the study of telomere lengths, but in particular, we're interested in them as sort of cellular markers of aging, but also as sentinels of stress exposure. And we know a little bit about the life course development in telomere length, and in particular that they seem to shorten very rapidly in very early life and then degrade more gradually across adulthood. But there's much sort of poorer or more limited understanding about what happens over the period of adolescence. And we would like to know this because biomarkers can be really valuable for studying things like health disparities or the biological embedding of stress exposures, even in younger populations who may not manifest other kinds of clinical markers of disease or disease risk, but biomarkers can capture that sort of underlying risk. So we want to be able to understand how we can apply these kinds of biomarkers to younger populations. I also want to just highlight a few other limitations of previous work. A lot of the work on telomere length has been done mostly in non-Hispanic white individuals, and in particular among adults. And so we have very few studies of telomere length in racially and ethnically diverse samples, and in particular in samples of children, and even fewer studies that have longitudinal data in telomere length in this particular demographic group and this developmental window. So why might we be interested in adolescent development, or this period of human development? We know that there's immense physical, cognitive, emotional, behavioral, and hormonal change across this period of development. We also know from previous work that the timing and pace of pubertal development is associated with adult health and aging outcomes, sort of at the other end of the life course. And adolescence is also a particularly promising developmental window into thinking through how early life exposures are connected to later life health outcomes. And so for these reasons, we think it's important to better understand the evolution and the change in telomeres across this period of development. So the goal for what I want to share with you today is really straightforward: to document and describe the cross-sectional and longitudinal patterns in telomere length across adolescent development and in a racially and ethnically diverse sample. The data I'll use are coming from the Fragile Families and Child Wellbeing study. I think a lot of you are familiar with this study, but just by way of very brief introduction, it's a probability sample of roughly 5,000 births in 20 large US cities, starting in 2000, following mothers and their children, and oftentimes fathers, across sort of early life and adolescence. It's a very diverse sample, majority minority, with 47% Black and 27% Hispanic. As the name might suggest, it's also a socioeconomically disadvantaged sample with a poverty ratio of about 25%, low educational attainment compared to the population. The analytic sample that I'll show you data on for today are coming from the year 9 and year 15 interviews. So the Fragile Families Study has followed children and their mothers primarily from birth at year 1, year 3, and year 5, and then drawn telomere length measurements at year 9 and year 15, and that's the data that I'll share with you today. There's also information on physical and pubertal development from both the year 9 and year 15 surveys. And so in terms of sample size, we have roughly 2,000 individuals at year 9 and at year 15, with a slightly smaller sample of individuals who have both times of data collection at 9 and 15, around 1,400. Dan and Lisa and colleagues have talked a lot about measurements, so I'll just briefly outline what we did here. It's telomere length derived from DNA saliva self-collection kits at both the year 9 and year 15 interviews. And what we have done is use a small subsample of year 9 sort of repeats to rerun them using the year 15 laboratory reagents and conditions, and then adjusted the year 9 samples using the average difference so that we have more confidence in comparing the year 9 and year 15 measurements. The measures of pubertal and adolescent development that I'll look at include chronological age. So year 9 and year 15 refer to the year of the sample collection, but there's a little bit of variation in the actual chronological age of the respondents during those survey years. We'll look at pubertal development measures at year 9, in particular a measure of physical development, which was answered by the primary caregiver of the development of the child relative to their peers. Everything in the developmental measures is scaled from less to more developed. There's also a summary score using measures of physical growth, hair growth, acne and other physical development changes measured at year 9. Then at year 15, there's a similar relative development measure, as well as for females, a report of age at menarche. Finally, we have body mass index measured at year 9 and 15, as well as polygenic scores for BMI, for height, and for age at menarche. Okay, so to get into some of the demographic patterns that we find here, what I'm showing you is the cross-sectional averages of telomere length at year 9 on the left and year 15 on the right, separately by sex and by race ethnicity. And we do see what has been pretty well-established patterns, demographic patterns in telomere length, with longer telomeres among females compared to males, and longer telomeres among non-Hispanic Blacks compared to non-Hispanic whites. But what I also want to draw your attention to is that on average, we see telomeres are longer at year 15 compared to year 9. Here I'm showing you telomere length at year 9 and on the left and year 15 on the right, plotted against the chronological age measured at the survey year. And what I want to point out is that we find no relationship between telomere length and chronological age in the cross section. There is less variation in chronological age at year 9, with pretty much just a 1.5 year window, but a little bit more at year 15. And we really find no evidence here in the cross section that telomere length is associated with chronological age. One thing that I'm really excited to get people's feedback on is this finding here, which is on average over adolescence, the telomeres are lengthening, but we see a lot of variation. So here we have telomere length at 9 and telomere length at year 15. for those who have both measurements, and the line where they would be equivalent is plotted here in red. And so everybody sort of above the line is demonstrating telomere lengthening over adolescence, and those below the line are demonstrating telomere shortening over this period of adolescence. And so perhaps contrary to what we might expect from the aging literature, we actually see a lot of telomere lengthening across this developmental period. To look at the measures of pubertal development here, I have the regression coefficients from linear regression estimates of sex stratified models of telomere length at year 9. And looking at the demographic measures, the developmental measures, and then the polygenic scores. A lot of null findings here, but a couple interesting things to note in terms of the polygenic scores, but only for females. We see higher polygenic scores for BMI associated with shorter telomere length, and higher polygenic scores here for earlier age at menarche associated with longer telomere length. That pattern is consistent when we look at telomere length at year 15 in the cross section. Although the polygenic scores are in the same direction for females, they're not statistically significant. And here actually at the year 15 cross section, we don't see any statistically significant associations with telomere length across the demographic, developmental, and polygenic score measures. And finally, looking at the longitudinal sample, so these are individuals who have measurements at both year 9 and year 15, looking at change in telomere length. So again, on average, we see a lengthening in telomeres. What we find here is that for males, there's some evidence that faster relative development at year 9, so they're developing more quickly than their peers at year 9, is associated with actually lengthening telomeres between 9 and 15. And here for females, the polygenic scores for BMI and menarche are actually associated in the opposite direction as they were in the cross section. So I'm curious to hear your thoughts on that. Just a couple notes on the robustness check. So we've run these things with a lot of alternative measures of physical development, looking at growth and height across this period, change in weight, change in BMI, and in particular looking at girls who experience very early puberty. The findings are substantively similar. We've also looked at some different specifications of telomere length, considering standardized telomere length, looking at categorical descriptions of change in telomere length. So just at those who are shortening, at those who are lengthening, or shortening or lengthening by some threshold here, 10%, as well as looking at percent change in telomere length. This is all to say that the findings that I shared with you are relatively unchanged, looking at all of these different specifications. So to summarize, in both the cross-section and longitudinally, we see that telomeres are on average longer at year 15 than they are at year 9. There's no association that we can document with chronological age and telomere length. And in particular, the pubertal and physical development measures are not consistently associated with telomere length in cross-section or longitudinally. And I think that this is really important before we get into some of the other substantive work that we're really interested in doing with telomeres, particularly in children and in adolescents, to think more about how we can use telomeres in this developmental period and how we might expect them to behave across this stage in the life course. I'd just like to acknowledge the co-authors on this work with me, a lot of them who are on this call, as well as our funding from NIH and NICHD, and I look forward to your questions.
Thank you, Lauren. We do have about 5 minutes per question, so feel free to use the chat or Q&A or hand raising, whichever you prefer.
I have a question. Very interesting findings. Surprising to see lengthening. And was this in the saliva samples?
It was, yes.
Okay. So I just was curious if you thought about like sort of the hormones that might be driving that. If we have, you know, activation of telomerase, for example, and maybe you could talk a little bit about what factors you think could be influencing that.
Yeah, I definitely think that some of the hormonal and biological changes that are occurring across the course of normal pubertal development might play a role here. We don't have ways to sort of get at those particular mechanisms that might be driving some of the findings that we have with respect to the lengthening of the telomeres. Yeah, I'm curious, you know, it would be really nice. One thing that's a little bit unfortunate is that the window of pubertal development that we have here is really, you know, at 9, those who have started pubertal development would be very early. And by year 15, most of the respondents have kind of completed a lot of their pubertal development. So it would be great to have a little bit more fine-grained information about their pacing of development over this period. And unfortunately, we don't have anything to capture that, although the age at menarche variable for females would capture a little bit more of that variation, and don't see anything there particularly related to those who are having sort of very early development. Dan, did you have anything that you would want to add there?
Well, yeah, thank you, Lauren. And that was a very nice summary. I haven't heard you put that whole thing together like that. It was really interesting. So I think that we have to look at this data in a couple of perspectives. First, it's over a relatively short period of time. Let's just say 9 to 15 or 16 years. Second, there is a variance to the accuracy of the telomere method. Mm-hmm. And third, you know, I wonder if the best conclusion, looking at it longitudinally, is that the precision of our assay is not such that we can reliably see differences in telomere length over that short an interval at this point in the life cycle. And that we're really looking at is regression to the mean, because unless I'm wrong, we're seeing about half the samples go up and half the samples go down. And the average telomere length change is actually very small. Am I right?
Yeah, that's right. If we are looking at this plot here, right, there's not really, on average, there's not that much change.
So now all the other things that people have mentioned, it could be that testosterone and estrogen, for example, really do affect telomerase activity in the stem cell populations that to a certain extent are populating the lineages that we're looking at. That's true, and we have evidence in our lab that might be happening. But it also might be that we're seeing a shift in the differential counts across puberty as people are maturing and moving from a polymorphonuclear dominant to a lymphocyte dominant. So I think all of these are variables that we're not accounting for here.
This is Stacy. So just feeding into the sort of what you guys had brought up before in terms of when these were extracted. Are we concerned at all about the time length that these were sitting before extraction? And did you consider that? Or did we look at the DNA integrity?
Yeah, so
specifically?
Stacey, I'm sorry, go ahead, finish your question, I'll answer that.
Yeah, so that was one. And then the other one is, and your analysis, I couldn't quite wrap my head around of all the things you did for the last slide. And so my other question was, given that we know that there are certainly differences in both the timing of pubertal onset and pubertal tempo between races, when you look at these within race, are you able to pick up a better association between puberty and or age? I am really kind of surprised that you don't see a correlation with age, because even in our toddlers, we're seeing a correlation with age. So I'm a little bit anxious about that. But I think that forgive me because I don't know what your sample population breakout size is within it, but I think, you know, those are sort of three thoughts.
So let me respond to the first, and then Lauren, I think, is better positioned to answer the others. I'm not worried about DNA integrity. We don't measure the integrity of every one of these samples, although we do when we're suspicious about it, or we did. Now I think we would do a bioanalyzer on all of these, as I was saying. What I can say is that these samples were collected by Westat, shipped in batches to my lab, and extracted promptly, very promptly, and put to -80. So I don't think that's the issue. And when we've gone back and looked, it hasn't been the issue. So just that's the best I can say for that, unless Lisa has a comment. No. Yeah.
No.
The other questions are good questions. I'll let Lauren address
them. Particularly, thank you for your questions with respect to the race stratified estimates. So the short answer is no, we don't find any stronger signal, even if we look within self-reported race ethnic groups. And I don't know, you know, that you need to be too worried with the contrast between what we're finding and maybe what you found in the toddlers. I think that, you know, this is a very sort of distinct developmental period. And so I don't know that we necessarily have, you know, too much reason to suspect that what we see in young children will be the same as what we see, you know, in adolescents. So I think it's an open question. But the point with respect to sort of differences in pacing and timing of pubertal development across racial ethnic groups, I think is a great one. But we don't find any substantively different findings when we look just within self-reported race ethnic groups.
Also, Stacy, just as a follow-up, you know, I'm obviously aware of that data you just mentioned, and when we look at greater intervals or different intervals in the lifespan, either in adults or in younger children, we see the expected decrease in telomere length with secular change, you know, with changes in age. I think that this finding here is related to several things, including the heterogeneity of the sample, the relatively narrow ages, and remember the age 16 was not at exactly 16, and the unmeasured variables that we're not accounting for, such as well, I'll leave it at that. I think that's the point.
We have a couple of questions here. They may follow right on what has been discussed and maybe they repeat it, but Shahina, do you want to ask your question?
Yes, so actually it's, again, it's very related to Dan's comment and Stacy's comment. First, it's an amazing work, amazing resource, but there are several things that stopped me a little bit. The age correlation was one. It's again, I agree with Dan that's possible of the small, like the variation in age is not that big, so you can see age correlation. But the second that is related to that is the lengthening of 50%, again, stopped me. And the third is that by any mean related to the saliva use. And one other thing that also, perhaps I will bring it later, is that telomere length KB distribution, where what you see is a little bit less than what in that age group, but that what you see with southern blot and flowfish. And I definitely understand that this is the qBCR absolute length. So again, it's not exactly the same. What I want to suggest perhaps, and as you know, there are a lot of argument now that the lengthening, as Dan's have alluded to, is just an artifact in a way. So I was wondering if you repeat some of those analyses in only those individuals or those children that shows, you just exclude the lens name and see what you see. Do you see the age correlation? Do you see, you know, all the other things you expect that we usually use as a marker of kind of an indirect QC marker of the method. Again, it's perhaps the saliva, you know, is causing some of those things that you see, and it's just returned back to the assay in using saliva and other things.
Right. Yeah, no, those are really important considerations. I'll address, I guess, your last point, which is curiosity about what happens if we look at just the people who are shortening from 9 to 15. And, you know, just as a kind of reminder, right, that's discarding almost half of the sample because they don't conform to sort of what we might expect over this developmental period. I'm a little bit hesitant to do that, but we did, I think, for the exact reason that you're suggesting. You know, we find well-established relationships in terms of sex and race ethnicity, but again, we're not finding over this short period of age variation, right, from about 15 to 18 in the respondents. You know, no relationship with age, even among those who are sort of only among those who are shortening. So, you know, I think it's certainly a puzzle, but it's important to understand or to document at least these patterns that we're finding here in the data.
Perhaps it's not strange, by the way, because now I'm sorry, I didn't, apparently I didn't know that it's only 15 to 18, but given even the correlation with age in the larger age distribution, when you see, you know, maximum perhaps 30% or so. So, you know, in this very, very small age variation, perhaps there is nothing. But I was just more concerned about, you know, things related to saliva and quality, and kind of now I feel better in a way.
We have another question from Diane Coffey.
Hi, Lauren. Thanks so much for this really interesting presentation. It sort of sounds like things are coming together around these measurement issues that the presenters have been talking about, and perhaps that's really what's at play. But I wondered if you thought about power as a possible explanation for the null results in your cross-sectional associations.
Yeah, that's a great point. I'm reading your question in here as well. So there is variation in you know, especially the measures of pubertal development at year 9, as well as age at menarche and some of the other measures that we're looking at, BMI, for example. So, you know, I'm not too worried about not having a lot of variation in the measures that we're looking at. In terms of power, I mean, this, on the scale of sort of traditional telomere length studies, this is very large, right? I mean, we have like 2,000 respondents at year 9, about 1,400 longitudinally. And so this is a really large sample comparatively, I think, to some early studies of telomere length in particular. So that's not to say that, you know, power can't be an issue, but I think that given previous work in smaller samples that find, you know, other kinds of relationships, you know, I'm not quite ready to say that that's the only reason why we're not finding particular associations, but it's a great point.
Stacy, do you want to make your point that you've put in the chat in person?
Sure. I mean, I would just say that we have a non-human primate study with Mara Sanchez out of the Yerkes non-human primate, and we actually so we measured the telomere length out to age, I want to say it would be age 16, the equivalent, and we saw a non-linear pattern. So overall decline, but we had repeated measures. So it was sort of the almost sinusoidal, but if you just sort of looked at the slope overall, it went down. But certainly it was nonlinear. And so I think I'm wondering what's happening, and you know, we can't go back and get the DNA, but I am intrigued by this idea that there's not necessarily a linear change, and that with only 2 time points, we're really missing part of the story.
Mm-hmm.
And, you know, again, I'm very fascinated by this, because while we have sort of estimates of expected rates of change in terms of telomere length in the adult population, we know 0 about the 0 to 20 range. And so I think this is super important data in terms of that. And I think there's just a lot that we don't know.
Yeah, thanks. I appreciate that point, especially, you know, we only have 2 time points. And so I'm curious to see, you know, what you're talking about in the non-human primate work. And I wish we had more repeated measures across this period that it's really a sort of volatile developmental time, right? So excellent, thank you for that.
And just for, if you guys want, I did have, we have done a power analysis looking at sort of the power and the impact on power for repeated measures telomere analysis with variation based on the ICCs. So we're sort of advocating from the TRN standpoint not to use CVs because they are somewhat impacted by where you start on the intercept line, but to rightly use ICCs. And so Simon actually did a power analysis to sort of say, if you're looking for a 250 base pair difference, what the impact is in terms of ICCs. And so the tighter your ICCs, so speaking to what Dan had said earlier, you know, the tighter that is, the better chance you have, but there's a huge impact in terms of your assay ICCs on the power to be able to really understand what's happening in terms of change. But I also want to highlight Dan's point that there's so much unknown and not controlled for in this high-risk population that could have their own impacts as well. So I think that's an interesting caveat.
Shahina has one more point to make.
Genomic
Yeah.
I'm not entirely sure that I know what you are.
Sorry. So using polygenic risk scores of telomere length SNPs, particularly here when you are looking at young individuals in relation to measure telomere length. Have you done that? I might have missed it. I'm sorry if I did.
I didn't miss it. That's a great suggestion. It's something that's on my list. We didn't have those scores yet for the sample, but yeah, it's a great suggestion. Thank you.
No more questions?
Thank you, everyone.
Thank you.
Eileen, what would you like to do? Would you like to go to our final question here?
We don't have to just keep working. I think we had a great discussion, and I think this was, you know, a really coherent session, and everybody learned a lot, and we have a lot more to do. And I think that this is basically the end of what we had planned and were able to resurrect for April. And I think we can move on. And thank everybody for attending. And I will note again that at our last week's session, the issue came up of measuring COVID antibodies in populations, and that people really want to have a session on that topic. So if you have suggestions, we know several labs that are working on things for large population studies. But if you have suggestions for good participants in that session, please let us know. It'll probably be sometime in August. We need to schedule around the fact that some people actually would like to take a couple days off this year, which seems like something most people are not planning really. But we are likely to have something in August about measuring COVID antibodies, although Some labs are still working on it because they've been allowed to open up to do this. And you know, we're waiting for results from at least 2 labs before we can schedule. So I think we can say from the Biomarker Network that we're pleased, and this does seem to work. We're able to keep connections in spite of the fact that we weren't able to get together in person. Teresa, do you have anything more?
No, just I thought it went really, really well. I'm really glad that we were able to do this. And as you say, Eileen, I think seeing how well this format works, it does open that opportunity going forward. So I think the one in August, and then we can see what other topics may arise that people would like to have a sort of remote virtual meeting to take advantage of being able to get together via that mechanism. So this is great. It's really encouraging.
And we are open to other topics. So if people want to write and say, let's have a topic and you want to organize it, great. And this is not a closed shop in any way. Anybody can suggest, and we thank the groups that the people in the groups that presented today, and I feel like we're making some progress.
Yes, absolutely. Thank you, everyone.
Thank you so much for organizing.
Thanks, Eileen, Steve, and Teresa. Thanks very much.
Oh, thank you, guys.
Our pleasure.
This is great.
Yeah.
Take care.
We're happy.
Bye.
Okay. Bye-bye.
Bye.